Bismillahirrahmanirrahim. In the name of Allah, the most merciful and the most beneficent. Today we will discuss about endoplasmic reticulum and what is endoplasmic reticulum, how it works, what are the main functions of endoplasmic reticulum, and basic structure of endoplasmic reticulum. Usually, it is considered as the budded structure from nucleus or nuclear membrane. So it has a smooth endoplasmic reticulum and rough endoplasmic reticulum. Rough endoplasmic reticulum usually have ribosomes on their upper side, so that is they are known as rough endoplasmic reticulum. However, smooth endoplasmic reticulum do not have ribosomes, so it is attached with nucleus. So in the year 1945, a lace-like structure was observed and named as endoplasmic reticulum, first observed in cytoplasm of chick embryo cells. So these membranes have different types of channels and different types of networks that is just attached with nucleus and present in cytoplasm. So these are single membrane cell organelles. These are different. These are interconnected networks of tubules, vesicles, and cisternae within the cell. So all these network of tubules, vesicles, and cisternae just collectively known as endoplasmic reticulum. So endoplasmic reticulum are considered as one of the components of cytoskeleton that just give a specific shape to the cell, along with microtubules, microfilaments, and intermediate filaments. So the term endoplasmic reticulum was first proposed by Professor Porter and Fullen in 1952. So it is present in the cell, and it is present almost in all types of eukaryotic cells. Endoplasmic reticulum present in cytoplasm and attached with the nucleus. So these are found to be absent in mature erythrocytes, ova, embryonic cells, and different prokaryotic cells. The number of endoplasmic reticulum just varies in amount from different types of cells. In spermatocytes, it is represented by a few vacuoles only. However, in the cell of adipose tissues, it is quite simple, having the form of a few tubules. The cells that are actively synthesizing proteins, including liver and pancreatic cells and fibroblasts, have large number of endoplasmic reticulum present in their cells of these specific type of cells. Endoplasmic reticulum forms almost 30 to 60% of the total membrane of the cell. So what is the basic origin of endoplasmic reticulum? There are different theories available, different hypotheses are available. However, the most widely accepted hypothesis is that endoplasmic reticulum is budded off from the nuclear envelope. Endoplasmic reticulum appears to arise from the outer membrane of the nuclear envelope by outfolding, or from the plasma membrane by infolding. You can have a look from the figures, then you will understand more accurately that what are the outfolding of endoplasmic reticulum and what positions are the infolding positions of the endoplasmic reticulum. The smooth endoplasmic reticulum seems to rise from the rough endoplasmic reticulum, and there is no ribosomes on smooth endoplasmic reticulum. So here we have a figure of endoplasmic reticulum. There are two basic morphological types of endoplasmic reticulum that we already discussed, that rough endoplasmic reticulum and smooth endoplasmic reticulum. Endoplasmic reticulum membrane is thinner to 50 Armstrong than that of plasma membrane, that is of 80 to 100 Armstrong thick. So here we can have a look to this. As we already discussed, that it is attached with the nuclear membrane and considered as the budding off of nucleus. So here we have this one in dark blue color, or you can say, or this what blue type color this is. So this is the rough endoplasmic reticulum that has ribosomes, and these are the smooth endoplasmic reticulum. These are the ribosomes, these are the cisternae, and tubules are also there. So the physical structure of endoplasmic reticulum in three-dimensional network of intracellular has three different parts, that is known as vesicles, tubules, and cisternae. Cisternae are sac-like elements, unbranched and flattened, They lie in stacks parallel to one another. They bear ribosomes on the surface, and because of these ribosomes, this is known as rough endoplasmic reticulum. Then there are different tubules are available, that is irregular branching element which form a network along with the other elements. These are often free of ribosomes. The third part is vesicles. The vesicles are oval and rounded and free of ribosomes like tubules. However, cisternae has ribosomes on their surface. All the elements of endoplasmic reticulum freely communicates with one another to perform different functions and contain a specific type of fluid that is known as endoplasmic matrix. These matrix is different from cytoplasmic matrix, as cytoplasmic matrix has different compositions and chemicals. However, endoplasmic matrix that is present inside the fluid present inside the endoplasmic reticulum is has different chemicals and different composition. The endoplasmic reticulum may pass from one cell to another through the plasmodesmata in the form of desmotubules. So here we have another figure, that this is the nucleus, that is nuclear pore, then there is a nuclear surface, nuclear envelope, then there is the budding off from the nucleus, that is known as endoplasmic reticulum. First we have rough endoplasmic reticulum that has ribosome, then we have smooth endoplasmic reticulum that don't have ribosome on their surface. These are known as cisternae that has specifically you can say ribosomes, and these are the cisternae space. What is the molecular structure of endoplasmic reticulum? The membrane of endoplasmic reticulum are composed of two layers of phospholipid molecules that are sandwiched by two layers of protein molecules like other membranes in the cell wall. So there are two different types of endoplasmic reticulum, that are rough endoplasmic reticulum and smooth endoplasmic reticulum. As we already studied about endoplasmic matrix, that is the fluid filled inside the cisternae space is known as endoplasmic matrix. The space inside the tubules and vesicles is filled with the watery type medium and material and a viscous solution that is different from the fluid that is already present in the cytoplasm or cytosol. This is known as endoplasmic matrix. Their walls are constructed of lipid bilayers membranes that contains large amount of proteins similar to the cell membrane. Then we have a smooth endoplasmic reticulum that do not have ribosome on their surface. So smooth endoplasmic reticulum is an arrangement of tubules, vesicles, and sacs. The size and structure of smooth endoplasmic reticulum varies depends on the different type of cells. However, smooth endoplasmic reticulum can change within a cell for lifetime to allow the cell to adapt to changes in its function and requirements. There are no ribosomes attached to the membrane surface, as we already discussed that the smooth endoplasmic reticulum do not have ribosomes on their surface. However, rough endoplasmic reticulum has. Smooth endoplasmic reticulum is connected to the nuclear envelope, as we already have a look to the figure, that both rough endoplasmic reticulum and smooth endoplasmic reticulum just attach with the nucleus and nuclear membrane and like a budding off of nucleus. The network of the smooth endoplasmic reticulum allows there to be enough surface area for the action or storage of key enzymes or the products of the enzymes. So this network has much space to just store a specific number of chemicals or enzymes that just perform different functions in the cell. So when there is a need of those enzymes and those chemicals, then the storage available inside the smooth endoplasmic reticulum, just use it to perform different functions. However, smooth endoplasmic reticulum is less stable. Smooth endoplasmic reticulum is characteristic of cells in which synthesis of non-protein substances takes place. So after smooth endoplasmic reticulum, we have rough endoplasmic reticulum. The surface of rough endoplasmic reticulum has ribosomes embedded on it, that is giving it a appearance of roughness, and because of that roughness, it is known as rough endoplasmic reticulum. Mainly consists of cisternae, as we already have a look to the figure, that the cisternae has ribosomes, and then there is a space in the cisternae that is known as cisternal space. The membrane of rough endoplasmic reticulum forms large double membrane sheets, which is located near and continues with the outer layer of the nuclear envelope. Rough endoplasmic reticulum is very important in the synthesis and packaging of different types of proteins, as it just make a packet or a macromolecule of the protein and just moves out those packet or macromolecules. Binding site of the ribosomes on the rough endoplasmic reticulum is the translocones. The ribosomes bound to the rough endoplasmic reticulum at any one time are not a stable part of this organelle's structure, because ribosomes are constantly being bound and released from the membrane. So all the time just ribosomes just attach and detach constantly. So sometimes the ribosome just release itself, and sometimes it just bound itself to the cisternal or cisternae of rough endoplasmic reticulum. Ribosomes only binds to the rough endoplasmic reticulum, and there is no space of binding of ribosomes to the smooth endoplasmic reticulum. Once a specific protein nucleic acid complex forms in the cytosol, ribosomes just attach itself to the rough endoplasmic reticulum. This specific complex forms when a free ribosome begins translating the messenger RNA of a protein destined for the secretory pathways. The first 5 to 30 amino acids polymerized encode a single peptide. That specific peptide just perform a specific molecular, just gives a specific molecular message that is recognized and bound by a single recognition particle, that is SRP. So in easy words, that the attachment and detachment of ribosome with the rough endoplasmic reticulum just depends on a specific protein nucleic acid complex forms in the cytosol. That specific complex just make the ribosome free when it start translating a messenger RNA of a protein for a secretory pathway. However, the first 5 to 30 amino acid just polymerized and uncode itself like a single peptide, or you can say very small protein or a peptide type structure that just gives a specific molecular message to for the recognition or communication with the other protein or other parts for a specific function, for a specific recognition, that is known as single recognition particle. The ribosomes that becomes attached to the endoplasmic reticulum synthesize all transmembrane proteins. However, most secreted proteins that are sorted in the Golgi apparatus, lysosomes and endosomes. So we already discussed about Golgi apparatus, what in actual Golgi apparatus are. Golgi apparatus also has tubules, vesicles and cisternae, and it also Golgi apparatus performs secretory functions, phosphorylation and different cell specific functions. Translation pauses and the ribosomes complex binds to the rough endoplasmic translocones. So what are the basic functions of rough endoplasmic reticulum? Here we have some key functions of rough endoplasmic reticulum, that are surface for ribosomes, because ribosomes just attach there and perform different functions and also translate the messenger RNA. So after that, surface for protein synthesis. This rough endoplasmic reticulum just gives space for the space and give different chemicals and helps for the synthesis of different types of proteins, formation of glycoproteins, synthesis of different precursors, and also the formation of smooth endoplasmic reticulum is only because of rough endoplasmic reticulum. So what are the main functions of smooth endoplasmic reticulum? That includes the function of lipid metabolism, carbohydrate metabolisms, and detoxification. It has specifically a function of abundancy in mammalian liver and gonad cells. It also synthesizes phospholipids. So, and rough endoplasmic reticulum just has a function in that includes the surface ribosome attachment, synthesis of different proteins, formation of smooth endoplasmic reticulum, and synthesis of different precursors. However, smooth endoplasmic reticulum just has a function of lipid metabolism, the breakdown of synthesis and breakdown of different lipid molecules, carbohydrate metabolisms, and also the detoxification of the cell is just because of smooth endoplasmic reticulum. What is detoxification? In actual, the function of detoxification performed by smooth endoplasmic reticulum that brings about the detoxification in liver, converts harmful materials, suppose drugs, poisons, into harmless ones for excretion by the cell. So after the smooth endoplasmic reticulum just make the harmful materials into less harmful means, so that the cell can excrete it easily and there will be no damage to the cell. Formation of organelles. Smooth endoplasmic reticulum produces Golgi apparatus, lysosomes, and vacuoles. Golgi apparatus just has different tubules, vesicles. Lysosome is just a package for that has different types of enzymes for the engulfment, and vacuole also. So it also carries out the attachment of receptors on cell membrane proteins and steroid metabolisms. In muscle cells, it regulates calcium ion concentration. The smooth endoplasmic reticulum also contains the enzyme glucose 6 phosphatase, which converts glucose 6 phosphatase to glucose, a step in glycogenesis. After that, now we have sarcoplasmic reticulum. Here we already studied about rough endoplasmic reticulum, smooth endoplasmic reticulum, function of rough endoplasmic reticulum and smooth endoplasmic reticulum. Now we have sarcoplasmic reticulum. Sarcoplasmic reticulum is in actual smooth endoplasmic reticulum found in smooth and striated muscles. The only structural difference between these organelle and smooth endoplasmic reticulum is the medley of proteins. Medley mean different types of proteins. Both smooth endoplasmic reticulum has different type of proteins and different functional proteins. However, sarcoplasmic reticulum has another group of proteins, another class of proteins, both bound to their membranes and drifting within the confines of their lumens. The fundamental difference is indicative of their function. So what they are just different. Sarcoplasmic reticulum is different from smooth endoplasmic reticulum because of different types of proteins and their function. The endoplasmic reticulum synthesizes molecules which the sarcoplasmic reticulum stores and pumps calcium ions. So here we have another figure that just explains everything. However, sarcoplasmic reticulum in the excitation contraction coupling in muscle cells are shown in this figure. So here we can see that this is the motor neuron muscle fiber, then just enlarge it and it has different activities here. So here in this yellow color just denotes the sarcoplasmic reticulum. An action potential black arrow arrives at the motor neuron terminal and vesicle of the arc are released. So these are the specific function, and that we already discussed that sarcoplasmic reticulum just synthesize different molecules and store it and then pumps the calcium ions out for the for different other functions. Thank you so much.